Hello, I'm Neil Spooner, and welcome today to today's PCSIG webinar. The title of today's presentation is Incorporating Patient-Centric Sampling, PCS, Approaches into Multi-Site Clinical Trials. What you're going to hear is the output from a workshop that the PCSIG held on the 11th and 12th of June 2024. But before we get started, I've just got a couple of housekeeping items. As always, the PCSIG wouldn't be able to do any of its wonderful work without the support of our sponsors. So thank you to all of these sponsors. And for anyone interested, the next sponsorship year is starting the 1st of November. So if you wish to sponsor PCSIG, please just contact me. We'll be having about 10 minutes at the end of the presentation for question and answers. Please use the chat function within here, and we'll try and get to all the questions we can. Please note the webinar is being recorded, and in a few days' time, it will be available to stream through both our YouTube channel and through our website. So we have a variety of speakers today. These speakers come from across the world, and we're all present at the workshop back in June, and acted as facilitators and or scribes at that event. So you're going to see a progression from myself to Chi, to Phil, to Alex, to Jen, to Dan, to Brenda, and back to me. So please be patient as we jump around a little bit, but we've got a lot of information to share with you. And as I say, the webinar will be available to stream for free after the event. So our event took place on June the 11th and 12th at the Roche facilities in Welwyn Garden City in the UK. And we had 48 attendees from 33 organizations, and you can see their logo here. And thank you so much to Julia for preparing all of these beautiful slides that you're going to see today. So what we were talking about was the clinical trial roadmap. And we split ourselves into various discussion groups looking at the role of the patients, the role of the regulators, and thinking about study planning, logistics, and study conduct. And what we talked about to begin with on day one was what were the challenges to implementing patient-centric sampling for multi-site and decentralized clinical trials. And then we spent day two looking at how we can overcome these challenges. And this is what we're going to be talking about today. Obviously, we're trying to squash 2 days of conversation into 1 hour's webinar. And you'll be shown later in the webinar where you can go and look for some of the content that came out of our workshop. You'll also get the opportunity to vote on what activities we should be prioritizing to move forward on next. So I'm going to finish speaking now, and I'm going to hand over to Chi. Thank you.
Thank you, Neil, and hi everyone. My name is Pakarinen, and I'm Head of Program Management Office for MediPace. We're a values-led patient involvement and research company, and PCSIG kindly invited us to join the workshop, but also to speak at the beginning to help set the scene by emphasizing the importance of patient involvement in life sciences, health research, medical product R&D, and we gave a kind of a peek into the evolving landscape and requirements. And I wanted to take just a quick minute here to reiterate what we said then, which is the importance of understanding why this needs to happen. And firstly, this isn't a nice to have anymore. Change is not only coming, it has begun. So there is a real requirement from regulators, ethics committees to have to have patients involved, patient needs, patient experience data integrated into studies, into the designs, into also involved in the design of the end product as well. Secondly, involving patients will benefit companies directly and help them in their efforts, because what's developed will address the needs and of the end users or the intended patient population. We also made a poster that kind of distilled this nicely so that everyone could take a copy of it and read when they have time or bring to their teams. This is also available on the PCSIG website, maybe. If not, then on MediPace. And we wanted to also emphasize that you don't have to invent anything. There are co-created guidances to help you reach out, engage, establish a relationship, build partnerships, and build trust to collaborate. The regulators are creating guidance and offer support to sponsors on how, when to collect and what type of insights or experience data to collect that is useful for them in the evaluation. So all is basically there. We just need to act. We participated in the workshop and facilitated the discussion around patient needs, and we focused on the challenges of practical involvement that people were facing, but also how patient insights might help and support the study planning. Planning and conduct phases. So we had a really lively discussion, and based on the first day discussions that were focusing mainly on challenges, we created a checklist. And the aim for this checklist was to really make patient involvement an essential part of clinical study planning early on, and to get sponsors to make a start before making it a behavior. So the checklist introduces 3 main themes. You can find this from the resource section when we get there. 3 main themes, and you can see them on this slide here. So we start from the center, putting the patient needs in the center and working our way out and integrating through all these other steps as well. Just quickly running through the checklist, the first theme is understanding and integrating needs and preferences through involving patients. So start early in your planning and consider also how you manage upfront the investments or process changes that you might need if the insights that you gathered might flag up something that you hadn't thought of first, for example. The second big theme is accommodate the needs to ensure accessibility, provide choices, provide flexibility, and purposefully also involve or engage caregivers, family, people from diverse backgrounds that's appropriate to your target population. Offer training, education, or support if needed in order to make the collaboration more accessible, but through that also the participation to your studies more accessible. And the last theme is managing change and expectations. Because processes might be rigid, so ensuring clear communication, making sure that you seek input and also integrate it to improve your study design, to inform your decisions, while also managing that there's the potential culture change needed within your team, but also beyond your team and partners. So this checklist was on the table the next day when we started talking about solutions, so that different groups could discuss and integrate patient involvement in their steps and when they were thinking about solutions and to their challenges. Thank you. Over to Phil.
Hi there. I'm Phil Garner. I'm co-founder of Pakarinen and a director of the PCSIG. On the study planning side of things, there was some excellent discussion over the 2 days. The first day looking at the challenges, and then the solutions on day 2. And during those discussions, some key points kept surfacing. So the primary challenges that we identified were around stakeholder engagement. So the need for education and persuasion of key stakeholders, like whether they are decision makers, budget holders, patients, that was considered critical, as well as also education of other cross-functional internal teams, basically to impart confidence in adding PCS as a new collection model into a study plan. And then the second key challenge identified was around protocol language itself and education on how to standardize wording around the use of PCS in the protocol, and what factors need to be considered. And this also would apply to the ICF as well. And what should we be including in there by default? Of course, there are other challenges identified as well, namely around data analysis, for example, how and when to conduct bridging or validation studies, around timing for engagement, in particular around early engagement of stakeholders and ensuring involvement of key teams like preclinical teams, for example, early on in the planning phase. And then also importantly around the vendor and device management, what assurance do we have, and how do we get that assurance that the device will be available, or if the vendor will still be there throughout the whole duration of a study, which could be over a long period of time. So day 2, we drilled into solutions for those primary challenges that we identified, and they kind of overlapped somewhat. On the stakeholder engagement side, it really came down to education and how we educate those stakeholders. So we talked about documentation, for example, providing appropriate FAQs or infographics, or even the use of decision trees to help to remove the perceived barriers, basically handle the objections that might come from different stakeholder groups who just aren't used to doing this sort of thing. So for example, it could be a FAQ document on the costs and cost structures involved with PCS, or the logistics, basically to just remove some of that fear. Then we looked at publications, so that could range from publishing peer-reviewed journal articles, or any kind of content on case studies of what worked, what didn't, lessons learned, to more specific things like regulator questions, not questions to regulators and their responses, but basically building up a body of evidence that can help convince and back up the need for these PCS approaches. And then thirdly, promotion, basically banging the drum of all this and how we do that, not only to ourselves who believe in this, but also to help to expose those who aren't already familiar with PCS. So that could be the use of PCSIG website, for example, as an information hub, and to provide lots of excellent insight and information there, knowledge there. Could also be to present like podium or poster presentations at key conferences, basically to get across what it is, why it is, and importantly how to do it in reality. And on the protocol language side of things, there's lots of similarities here. It all came down to information and education again, and we looked in particular at a decision tree type model, which perhaps could be accessed via some sort of interactive tool or website. Like for example, a user could input certain information about a study, like like is the device approved? Is the device you're using approved? What is the data being used for? Is it a study about the device itself, or is it about an indication? Geographical considerations, like are the devices that you want to use available in all the different locations that you need them to be? And is a sample is going to be taken centrally, or is it decentralized, or hybrid study? Anyway, they're examples, but there could be many of these different types of questions and parameters that are asked in this decision tree. And the output would be a series of templated documents, which would be useful start points. So for example, it could throw out this a template study plan, ICF, lab manual, but also a set of common FAQs for all different stakeholders, from your budget holders and key decision makers all the way through to the different teams and patients. And if done right, it can even provide those templates in the appropriate language to make it as accessible as possible for each of those stakeholder groups. And it's a huge bit of work, there's no denying that, but it would be extremely beneficial and a great way to standardize the approach, which would make it more efficient, and also it'd make it more familiar, which in itself would help to break down those barriers and support greater stakeholder engagement. Anyway, that's what we discussed. So I'll hand over to Alex.
In our country report, the Italy said that they're expecting a final, but we see this.
Great, thank you, Phil. Hello, everybody. I'm Alex Georgiou. I work in regulated bioanalysis and biosample operations at Roche in the UK. And very similar to Phil, we've got a study conduct section, as you would have seen on the clinical trial roadmap. There were 6 main categories that have been identified as important during study conduct using patient-centric sampling. And the 2 primary challenges were the sampling device and the matrix selection, and then data capture and integrity. So for device and matrix selection, we felt education was key to understanding the different options available. What is the best device? What is the best matrix combination based on your analyte data usage and patient population? For data capture, verification of accurate and GCP compliant data is needed to limit metadata queries and ensure data integrity, which is very important, as you all agree, at every step of the process. And then 4 additional challenges, which are our secondary challenges, were identified to be 1, patient compliance, 2, patient recruitment and retention, 3, sample analysis, and lastly, results and data reporting. So for patient compliance, we felt the need to ensure that the patient can and actually does collect the samples per instructions for viable samples and usable metadata and also results data. To make the patient recruitment, to make sure it's not impacted and that patients are actually retained for the study duration is important. So sites and patients need to understand and see the benefits of patient-centric sampling. And to link the patient-centric sampling to our sample analysis, we need to make sure that we've got information on the sample chain of custody, the limitations due to sample volume, have confidence that complex processing has been followed to meet the needs of our assay requirements. And then for results and data reporting, questions like what happens to the source of the data, how does it get back to the site investigator, how do we link all the information through the end-to-end data flow, and the whole reconciliation process needs to be well understood. And then so for the solutions for these challenges, shown on green on the slide, and some of them you'll see are the same as Phil spoke about, but obviously we're now answering slightly different questions in the study conduct phase. So firstly, one of the solutions was to have informative and accessible documentation for all, including a decision tree to aid the device and the matrix selection. So this could include variables like sample volume, patient choice, the patient population, because there might be certain devices that suit certain patient populations. There might be differences based on the therapy area and also the use of the data. Things like feasibility questionnaire for the sites and study teams could be useful aids, as well as a process map for decision making along the drug development pathway, and also a best practice guidance for assay validation. So we thought of things like developing a Wikipedia-like resource where we can consolidate all these resources and join them up along the whole chain of events.
But I think that's where it gets a little bit tricky.
So next, the publications and promotional materials. So these are needed on patient-centric sampling with an industry standard. How to bridge data was an important topic that came up. So for example, how do you bridge plasma to blood data or venous to capillary data, and also how to interpret it. In addition to that, maybe even how to avoid it altogether by early project planning. It's also important to note that there is already a wealth of existing information to be promoted on the PCSIG website, and solutions can also be sought by engagement with patient advisory groups. So always thinking about the patient and what's best for them in this process. In terms of assay compatibility with patient-centric sampling, there was the manufacturer engagement solution. So this was thought necessary to ensure forward thinking. So in analyzer design, for example, maybe we need to accommodate different tube types and reduce sample volumes to make it really viable to collect these patients and follow that through into the analysis. And also thinking about the devices themselves, how can they be adapted to expand the choice of available anticoagulants and stabilizers, and also to make sure they're approved for use in multiple regions so that they're truly global. Then the solutions for data capture and integrity focuses really on having the right technology to meet our needs for data recording. It was felt that one single solution was really important for all clinical sites to make it easier for them as well as the patients. It's important to increase adoption of patient-centric sampling, and we felt that making sure things were automated, digital, for example, using barcodes, pre-filled information, artificial intelligence, to name but a few, and also making sure the patient or caregiver experience was enhanced. So using prompts, alerts, reminders and instructions for when they're having to take their samples, and also to make it accessible, so in multiple languages. And we see maybe adopting existing solutions across many large companies that we might use on a day-to-day basis already, could be useful in this area. And it's also important to make sure that we have the technology accessible to everybody, or provide it to patients and caregivers without that access. And then finally, understanding the end user of the data types is important, and that usage of the metadata that's being collected, because we have to make sure that the technology is actually capturing what's required. And another point was raised that if we triage non-essential queries on our metadata and hold them for later reconciliation downstream, this can result in quicker results and data reporting. So that captures everything that we discussed on study conduct. So with that, I will hand over to Jen.
Thanks, Alex. And hi, everyone. So I'm Jen Russell. I work as a biosample project leader, and I help to facilitate the discussions around logistics. As I'm sure you can appreciate, there are a number of challenges, and like the other slide summaries, we've broken them down into primary and secondary. So the primary challenges that we covered were chain of custody. So how do we keep samples anonymous but still fully traceable? We need to be able to trace final data all the way back to the individual collection device that was used by the patient. And the second primary challenge was around the training and feedback. So how do we train the sites and enable the patients to recognise a successful collection? Some of the other challenges that we discussed were around sample and data integrity. So having global solutions available to account for the various temperatures and shipping times that we face across the globe. With the shipping and couriers, it's how do we ensure patient anonymity and and at the same time reduce the patient burden. So minimal sample processing requirements, no complicated packaging or shipping requirements. And then lastly, around the patient experience. So how do we keep the patients comfortable with this different collection method and make it as simple as possible for the patients? So if we move on to some of the solutions that we discussed for chain of custody, for the technology and relationships, we really should try to lean on existing technology and also relationships that we have already with our couriers and various other companies. It was mentioned that the use of e-requisitions can really help with multiple aspects of chain of custody. So this includes your labeling, the kit management, and that full traceability end to end. Further education we thought was needed, and again really to leverage what is available and what other industries do, so not to reinvent everything just for clinical trials. Some recommendations came to partner with the likes of Amazon or Google. They certainly seem to understand chain of custody with our shopping, so maybe they have systems and tools and things that we can leverage here. There were also other companies discussed who are working already in this space. So TrueLab and Slope were just some of the ones that were mentioned. But it would be great if the PCSIG and these companies could really come together to discuss the challenges that we felt were there, and there's numerous that can't quite fit on the slides, but also to help drive some of those solutions. Long term, we recommended standardization at the industry level for metadata naming conventions. So this could make the whole chain of custody tracking so much easier. From QR barcode scanning, it would allow automation for device and chip handling at all of the labs, and could really support a full digital chain of custody, which would benefit all of us. And then if we move on to solutions, or some of the solutions for around the training and feedback, we recommend for collections that all patients have their first collection with any patient sampling device at the site. This would allow them to overcome any anxieties that they might feel around the collection, processing and shipping, whilst having access to the study nurse and support in real time. For the training model, we would recommend train the trainer. So the manufacturer or the sponsor train the site, and they would then train the patients. It was also noted that this should be verified and documented so that we are confident that the patients understand the requirements and are able to collect these samples at home. We should also consider site support post-training, so ongoing check-ins with the patient. It was noted that sampling at home can sometimes feel isolating, so having those check-ins so that the patients feel that they can raise any questions that they may have with their study team. And it was also noted that this site training and buy-in is really key, and we need this site engagement so that the clinicians can accurately represent the option of having home collections to their patients. For the training materials, I think it's no different to a lot of the training materials we provide in that they should be clear. They should have visual instructions that are easy to follow and available online and in paper formats to suit everybody's needs. It should be layman's language and available in local languages as well through translations. We also recommended that within the training documentation that there are examples of good and bad collections. So what a clotted sample would look like, for example, and ways to troubleshoot this and try to make a better collection next time. Part of the training that we helped with what that we thought would help the patients in really understanding the importance of collecting the samples, following the process accurately, and recording their metadata accurately. We should try not to overwhelm the patients with the scientific information, but I think giving them the understanding of why this is so important could be key to having successful sample collections. And finally, on this piece, it was feedback on the collections. So we felt that patients would only really hear back if their samples that they collected were not of good quality, but it is important to make them feel part of it, and perhaps giving them a notification that their sample was received and it was in good condition. So, you know, great job, keep doing as you are. As I say, there was a lot more discussed, and a lot of it can be found on the PCSIG site, but for now, I'll pass over to Dan to cover the regulatory engagement.
Great, thanks, Jen. So hi everyone, I'm Dan Baker. I'm a bioanalytical scientist currently working at AstraZeneca in Cambridge. So I helped facilitate and summarize some of this discussion around regulatory engagement. And there were two kind of main questions around regulatory engagement. One was around how and when do I engage and what sort of data I need. And then there was another around burden of proof and kind of responsibility of data and ensuring data quality. So when it came to that initial question of how and when do I engage and what data do I need, there was actually quite a diverse school of thought across attendees. So one school of thought was sort of saying, you know, let's do a lot internally beforehand. Let's do a validation. Let's build a strategy around PCS, plan out bridging, when it will happen, what it would look like. Conversely, another school of thought was sort of saying, let's do a small amount of work to kind of show a proof of principle, and then approach really early with regulators and sort of say, you know, before we sink all these resources into validating this, will this be a problem? Versus yet another school of thought, which sort of said, you know, from a PK BA perspective, we're covered by ICH M10. We don't need to do anything more. So they were quite diverse and different opinions that kind of floated up there. And I think some of that was reflected in the type of endpoint we'd be looking at. So generally, people seem pretty comfortable with ICH M10 for BA validation. When it came to things like companion diagnostics or sort of safety panels, etc., there was a little bit more hesitancy and perhaps a lack of clarity around what kind of engagement or documentation or data packages might look like in that area. One thing which was shared, which kept coming up, was sort of a unified strategy approach internally. And I think Phil's touched upon this already. You know, bringing in bioanalytical colleagues, PK modelers, operational, etc., building a kind of a unified strategy. Either whether you then go on and approach regulators with this or not, you've got clear and concise messaging around how you're engaging. The idea being that an organization is presenting sort of a unified front around PCS, supported by good documentation, resulting in hopefully fewer questions or pushback. One thing which was flagged up was sort of the power of the patient for stakeholder buy-in, I think, as Chi has already spoken about as well. And quite a few attendees sort of referenced that when the patient wants a microsampling approach to happen or a patient-centric sampling approach to happen, there's generally more push internally to support this and support the generation of evidence for regulators that this is a strategy just wanted and that will sort of have strong benefits. And there was some discussion that the regulators look favorably upon having that patient voice when designing a sampling strategy to help push them to just kind of accept that this is an approach that's worthwhile. And it was suggested that I think PCSIG could play a part in this by perhaps acting as a conduit for sort of patients' voices. And I think there are some documents on the website already which sets out some ideas around that. There was a note that did come up around the heterogeneity of regulatory bodies. You know, they do vary by therapy area, by department, by person you're engaging with. They're not necessarily homogeneous in some respects. But I think the key message that kept coming up was that we need to try and make them think this isn't the first time that this is being done. So not letting them feel like this is a new thing. So I think the idea was PCSIG could again potentially engage in this area with either decision trees or templates. And I think again, some of that is already filtering through onto the website. Or examples of use cases of successful implementation of sort of a PCS approach in a clinical trial, or potentially even acting as a conduit for sort of multi-company cross-industry viewpoints collectively saying that we as an industry kind of want to try this approach. We're unified and it's going to work in a similar way. It's not new, you know, it's nothing unusual. When it kind of came to data ownership and burden of proof, this broke down into discussions mostly around the different types of trials, sort of endpoints or sample endpoints which are available, or maybe used when using a PCS or microsampling approach, whether that's like a PK endpoint or device-based monitoring or stratification or something like a safety panel. And some discussion was had around where the analysis took place and who was responsible for looking after that, for that data. And they were kind of broken down into sort of those assays sort of defended by pharma, or those assays defended by an external organization, for example, say a device manufacturer or a clinical lab. And I think for PK and biomarker workflows, people seem to feel pretty happy that pharma had a good handle on this. For PK in particular, there was quite a lot of agreement that, you know, ICH M10 was enough and that people were happy that they're quite well covered with that. For things outside that, the situation became a little bit more complex. And I think there was some discussion around sort of assays and assay context of use. And it seemed to be that if an assay that had been developed and validated was being used within its context of use or intended purpose, then there'd be no sort of additional assessment needed. But then, you know, it could be used sort of off the shelf style. However, if there was something where an assay or device was being used outside this context of use, whether this might be, you know, a different therapy area or a different patient population, or, you know, venous versus capillary, then there may be some additional validation required. And I think this is where the collaboration aspect kind of come into play. I think, as was mentioned earlier as well, around these external organizations either producing these devices or generating these types of data, and pharma, you know, kind of working together that show that maybe a capillary approach is still acceptable. And I think this was generally an area where attendees kind of felt there were maybe some more questions that needed to be answered, and maybe there was a role for PCSIG to play in that, again, being some kind of cross-industry conduit. I think when it comes to data ownership and burden of proof, though, consensus generally seemed to be that whomever was sort of certifying that the data was okay and accurate is the one that's really responsible for defending it to the regulators. So there were really quite some varied opinions around engagement. However, I think the key thing was clear, consistent messaging, good documentation, and sort of a unified strategy internally, alongside the power of kind of amplifying patient voices to help kind of give more backing to what you're doing when you're engaging with the regulators. So with that, I will hand over to Brenda.
Years with specimen management experience across the drug development chain at Pfizer, Merck, and BMS. And based on that, I founded Clinical Transformation Partners, a biospecimen operations consulting firm. I'm really pleased to be here today to talk to you about this important topic. So let's pull all these threads together. We are at an inflection point in clinical trials. With the shift in emphasis from the sponsor to the patient, as well as the emphasis to expand access to trials to traditionally disenfranchised groups and wider geographic areas, the arrival of patient-centric sampling devices could not be more promising. What is more patient-centric than allowing a patient to collect a sample within his or her own home versus traveling for miles or even hours away and routinely engaging them for feedback on how the process can be improved? Yes, the process, because it's not just about the devices, although the devices themselves are a huge innovative step forward. It's also about analyzing the impacts of using these devices across the clinical trial process and including patient, sponsor, site, manufacturer, and regulator considerations at every step. As my colleagues have just discussed, we focused on 4 pillars: study planning, study conduct, logistics, and regulatory engagement. From a study planning perspective, stakeholder engagement is key, and one way of achieving that is getting doctors and patients buy-in and using that to influence internal teams who may just want to continue with the status quo. But once there's buy-in, One must alter the language in the protocol and the study manual to reflect these changes that respect and include the patient experience. Patient experience and options for the patients are paramount to success. We discussed building tables listing device options and having logic trees to help people make decisions regarding device and matrix selection, and understanding the challenges and possible solutions for data capture to ensure both privacy and data integrity. We discussed logistics. Oh, the many steps and challenges associated with logistics. The challenge of chain of custody, from sending and receiving the kit to time date stamping the sample collected, to understanding the quality of the sample from a patient perspective, educating them and making them aware, to tracking where the sample is at every point and what temperature it was on its journey to the testing lab to ensure data integrity, and the need to have industry standards and technology developed to do this chain of custody tracking. Finally, we discussed regulatory strategy and the need to have a map of global regulations and then maintain a database of these regulations and how they change over time, so that both manufacturers and sponsors understand the various regulations under which devices may be used across the globe. This in turn influences engagement strategy with regulators and is a key component to optimizing things for success. So how do we practically implement this? Well, if you look at this slide and around the margins, you see that we implement this through collaboration with patients, sponsors, sites, manufacturers, and regulators. Manufacturers and sponsors work together to devise a strategy, work together to raise awareness and educate our partners in the clinical trial ecosystem through our publications, our templates, our proposed protocol and study manual language, and promotion of these ideas. With this engagement and partnership, we can co-create training models and training materials that do take all perspectives into consideration and are patient-centric, as we work together to devise data requirements for the device technology and chain of custody. All of this can be found on our website. Next slide, please. So by scanning this QR code, you can see the readout from the report of our workshop, as well as access the patient checklist, the protocol inserts, and study manual inserts that we devised. And now I will turn this over to Neil to discuss next steps.
Thank you, Brenda. So what we'd like to do next, as hopefully you've seen from all the presentations today, and a very brief summary of 2 days of very in-depth discussion, we're now looking at what can we do to help organizations who may be interested in patient-centric sampling, but are a bit nervous about implementing it. As Brenda showed on the last slide, we've started to build some content on the patient-centric sampling interest group that people can go to on our website that will help guide them through, help hold their hands, help share experience from other people. But as you heard from all the presentations, there's a lot of things we could do. But obviously the patient-centric sampling interest group can't do all of them. So what we'd like to do now is ask you to vote on what you think is the priority we should be working on. We'll also be placing this survey on our LinkedIn group in the next few days. And I'll show you a QR code too, if you're not already part of our LinkedIn group. I'll share with you a QR code later that will take you through to that, and you'll be able to vote there as well. But basically, as you heard, there's a bunch of things we could do. We want your vote to understand what we should do. And also, most importantly, the patient-centric sampling interest group isn't able to achieve anything without all of you putting up your hands and saying you're willing to give up some of your time to help. Most of our teams meet, say, once a month for an hour. It's not a big commitment of time, but it is a commitment of time, and we recognize that. But if you're willing to give your time, then please just drop us a line at neil@pcsig.org, or leave a comment on LinkedIn. and we'd love to have you involved in this team and in other teams that we've got going. So the things we're going to ask you to prioritize, it's difficult because there are many individual things, but we met in a post-conference, post-workshop meeting to just figure out putting some of these things into separate buckets. And we arrived at 4 separate buckets, and we'd probably all love to do all of these, but without lots of resources, we can't do them all. So one thing that we talked about a lot is maybe on our website, developing decision trees around selecting the correct sampling device, the correct matrix, maybe a decision tree on the clinical trial roadmap that would help you along the way selecting what you should be doing and who you should be engaging with along the way, including engagement with regulators. So that's one of the things you can vote for, the decision trees. Another big area was different engagements that patient-centric sampling interest group could make. For example, with different manufacturers of tubes, the devices, the automation solutions that maybe we need to help us analyze these samples. Maybe we need to engage with different logistics providers, as we talked about earlier, people such as Amazon and Google, companies such as Slope and TrueLab, who manage to do this, moving samples around every day. And another engagement is maybe we should work with standardization of metadata, naming conventions, and be engaging with patient groups. So that's the second vote you can make. The third one is on publications, people sharing their experiences of what has worked for them in patient-centric sampling, incorporating patient-centric sampling into their clinical trials, what kind of questions they've had from regulators, what kind of responses they gave, what kind of lessons they've learned, what a bridging design might look like for a bridging study, and whether it's necessary with certain designs in your clinical development program. And so the fourth thing to vote on is, should we be developing standardized training materials, whether that be written or video or other formats, developing them that other people can use, things that are in plain language, plenty of visuals, and in lots of different languages. So they're the things we're going to ask you to vote on. Phil, I think you're going to be hopefully able to put up, I don't know how to make the voting thing happen.
It's done. It was happening whilst you were speaking.
Okay, I didn't even see that. Thank you, Phil. Okay, is it closed now, Phil, and are we able to share the results of that?
It is closed. Yeah, I can share something. Although I put it up a couple of times because it kept expiring. So I think we'll be able to consolidate the results into one view. But I can. This shows the general trend. Which is about 60% for engagements,
okay,
and 40% for decision trees.
Okay, so nothing on publications and standardized training materials.
Well,
okay.
there were some. So as I said, I ran the poll a couple of times because it kept expiring, and there are some votes on the first two that for those, but it is mainly. Engagements wins out and decision trees second.
Okay, fabulous.
A long shot.
Thank you, Phil. So if we move on past that, thank you everyone for voting. I'd like to move us on to the Q&A session now that we've got that. But going back to the previous thing with the vote, we will be placing this vote onto our LinkedIn community as well. that'll be open for a couple of weeks for people to vote on there. We'll combine all the votes together, and based upon that, we'll go to the PCSIG board with a recommendation of what team we want to launch. And as I said, we'll be looking for people to step forwards, to take part in these different activities. And the more of you that step forwards, the more activities we're going to be able to do, and the quicker we'll be able to move forward this kind of content to be able to share on our website that will help support people that want to incorporate patient-centric sampling into their clinical trials. So I'm just looking whether there are any questions, and I don't see anything on the Q&A at the moment. Do any of the presenters have any questions at all? Okay. Well, we are, if people do have questions following on from this event, please send them through to us at neil@pcsig.org, and I will be able to pass them on to the various presenters, and we'll try and find a way of sharing that through either our LinkedIn group and or through the website. But I'm not missing any questions, am I, Phil?
No, you're not. No.
Okay. So I'll keep my eye on whether any questions occur, but I'll just push on a little bit. So for those of you that don't know about the Patient-Centric Sampling Interest Group, we're a not-for-profit bringing together a lot of interested parties who wish to develop and promote the use of patient-centric sampling technologies for blood plasma and other human matrices to better facilitate the advancement of human healthcare and well-being. And that's not just for clinical trials, it's for general healthcare, it's for drug testing in sport, therapeutic drug monitoring, wellness, anywhere where a blood sample is taken and where these alternative approaches well, it's not just blood sampling, it can be for urine as well and other samples, but any place that a biological sample is taken, that these approaches might be useful. So if you want to visit our website, please use the QR code on the left. If you wish to learn more about our LinkedIn group, which is where we talk to each other, and there's about 450 members on there now, please use the QR code on the right. Just so you know, some of the activities that we have an active webinar program. We have one webinar per month other than in the month of August. And we now have quite a lot of webinars built up on our website and on our YouTube channel, covering various aspects of the use of patient-centric sampling, looking at it in different ways from the point of view of the clinician, the patient, the analytical scientist, academics, a variety of different uses of these technologies. We have things like listing of different sampling technologies on our website. We have a new feature, which is our library, which for different applications and user groups has a bunch of readily accessible content, whether publications, webinars, etc., that may be of interest to you. And as we talked about earlier, we've now started to build a lot of content on PCS for clinical trials, and we'll be doing similar things for diagnostics and other applications for patient-centric sampling. Just give a little plug to our upcoming webinars. So later this month, on October the 22nd, we've got a webinar featuring opportunities and challenges in mobile phlebotomy and self-sampling for precision chemotherapy dosing in clinical trials, brought to you by a couple of presenters from Karolinska in Sweden, which I think is very exciting and very much looking forward to that. And in November, the PCSIG board will be making a presentation just giving a review of what we've achieved in 2024 and maybe giving a little bit of a peek into 2025. So with that said, I'd just like to thank all of our partners and sponsors again, without whose support we wouldn't be able to have these webinars and our other activities. But most importantly, I'd like to thank all of our presenters today for just presenting fabulous webinar, and particularly to Julia for producing such beautiful slides that we were all able to work from. And what a great team coming together from different organizations around the world, able to speak with one voice about how we can help each other in bringing patient-centric sampling to clinical trials. And importantly, obviously, thank you to all of you for attending and taking part in patient-centric sampling and interest group activities, and making our group such a dynamic place to be. And thank you, everyone.